Welcome. My name is Bill Mobley. I'm a neurologist here at UCSD, and I'm the director of the new Sanford Institute for Empathy and Compassion. Our institute aims to understand the brain basis of empathy and compassion and to understand further how we can enable people to harness more effectively their ability to function with empathy and to reach out with compassion. I'm really entertained and excited about the aim of the conference, which is to think about those technologies that allow us to understand and harness and harmonize the intersections of body, mind, and brain. And this session features that very prominently. Very excited about the talks we're going to hear about, the data we're going to learn about, and the speakers will speak for about 25 minutes. We'll have 5 minutes of questions, and would really eagerly encourage you to ask your questions. So the first speaker is Esra Tasali, University of Chicago, Sleep and Metabolic Health. Esra.
Good morning. Thank you very much. It's a pleasure to be here in San Diego, and I would like to also send my special thanks to Gert, who invited me to speak at this workshop today. So we heard a lot of fantastic talks yesterday on how brain works and somewhat related to the periphery, to the body. So we're now moving more into the body aspect of things. So before we start, I'm from University of Chicago. I've been there for 20 years. I'm a clinician, scientist, pulmonologist, sleep medicine specialist, and I also have endocrine training. Let me tell you a little bit about the sleep research, the history of sleep research at the University of Chicago. The world's first sleep laboratory was actually established by Nathaniel Kleitman at the University of Chicago in 1920s. And later on in 1953, the dream sleep, REM sleep, we call it rapid eye movement sleep, was discovered at the University of Chicago. And today, the way we score sleep, record sleep in some ways, have all been established, those techniques at the University of Chicago by researchers like William Dement, who was a trainee at the time, and Alan Rechtschaffen. And I am very grateful that 20 years ago, when I was a fellow, Alan Rechtschaffen, who was an emeritus professor, signed my, my manual, the manual scoring manual, that we still use today. So let's move on and have a little bit of background on sleep regulation. Sleep is regulated by two mechanisms. One is the circadian mechanism, and the other one is homeostatic mechanism. The circadian mechanism is orchestrated with what we call the master clock, which is in the suprachiasmatic nucleus in hypothalamus. And the major stimulus for master clock is the light-dark cycle. And master clock through neuronal pathways and hormonal pathways such as melatonin and cortisol sends input to the so-called peripheral clocks that are in the tissues. And initially it was thought that circadian clock orchestrates everything, but now we know that the tissues' metabolism are also driven by peripheral clocks themselves, and they can oscillate independently in liver, muscle, fat cells, and pancreas. And the homeostatic component of the regulation of sleep is like an hourglass mechanism. So the more we stay awake, the more we build up the sleep debt, sleep need, and then we pay it off by sleeping at night. And this is basically an example of if we were to study you in a sleep lab today, we would go ahead and score every 30 seconds of your sleep and assign a sleep score there. There are two main types of sleep, what we call slow wave sleep, the non-REM deep sleep, and then we also have the rapid eye movement sleep, and they have very different physiology. Usually a normal night starts with what we call the stage 2 sleep, sort of an intermediate state between the light sleep and the deeper sleep stages. Then we have a short REM cycle, and then these REM cycles get longer and longer in a typical night as we go on. So how is sleep is related to metabolism? This is a very nice experiment that's been done years ago, 2 decades ago now, in 1999. What we looked at the 24-hour profiles of various hormones, and cortisol, for example, is a hormone that is circadian dependent. This is an experiment where healthy volunteers slept here, no sleep here, and an abnormal daytime sleep here. As you can see, the cortisol follows his own diurnal rhythm regardless of sleep happened or not. So it's a very circadian dependent hormone. And another example is a sleep dependent hormone. It's growth hormone. So growth hormone, when sleep happens, there's the major pulse, especially in men, that coincides with slow wave sleep. And when there's no sleep, that pulse disappears or attenuates. And then when you do sleep at an abnormal circadian time during the daytime, you still have the big pulse. So it's very sleep dependent. Even my son knows now that he has to grow, he has to sleep. So what is going on in terms of the world? How are we sleeping? There's a major epidemic that, this is a map from CDC Center for Disease Control, that there is a sleep loss epidemic. In various states, they did a survey. This is self-reported sleep duration, and a majority of people reported that they were not getting enough sleep. And on the same time, there are also epidemics of metabolic diseases. Like we know obesity from 1994 to 2015, those are all those CDC maps. The darker the color, the more obese the people are. As you can see, we got darker and darker, redder and redder over the years. Similarly for diabetes. So these 3 epidemics are really major issues these days. So what do we mean by sleep loss? It can happen by various conditions. One of them could be circadian rhythm disorders like shift work, or insufficient sleep duration, habitually curtailing our sleeps for social reasons or work-related reasons, poor sleep quality, obstructive sleep apnea, one of the most common sleep disorders, which we're going to talk about, and some other sleep disorders like insomnia, restless leg syndrome, or other neurological diseases. I will focus on the restricted sleep duration, poor sleep quality, and sleep apnea today. So what are the potential mechanisms that can link sleep loss to diabetes and obesity? There could be alterations in brain glucose utilization. After all, definitely sleep loss affects the brain. There could be through what we call HPA axis, hypothalamic pituitary axis, through cortisol release, through autonomic nervous system activity. You heard about the sympathetic, parasympathetic balance, energy balance, food intake and energy expenditure, or fat cells themselves, adipokines, the inflammatory pathways, and released molecules from the fat tissue, and free fatty acids. Free fatty acids have been shown actually to really play an important role in causing insulin resistance and diabetes. Insulin is the hormone that helps our blood sugar to be disposed to the body. So let's give some examples of each of these different components. Can sleep restriction impair glucose metabolism? I don't have time to go through all the studies, but this is just to tell you that over the past 2 decades, starting with the first study in 1999 published in Lancet in our laboratory, there has been a plethora of various designs and various studies in different labs using an experimental manipulation of sleep timing, duration, and quality using glucose challenge tests. We basically give glucose either IV or orally and look at how the body responds. And the conclusion from these studies is that there is now, we know, a convincing evidence that short sleep duration or disrupted sleep reduces insulin sensitivity, make people insulin resistant, and impairs glucose tolerance and increases diabetes risk. Let's give you an example of some of the protocols that we do in our laboratory or have done. We basically take healthy volunteers, give them in this particular protocol 4 hours of 8.5 hours bedtime, 4 days of 8.5 hours bedtime. And in another occasion, a month apart, we also do in random order bedtimes of 4.5 hours. And during this time, we do sample various hormones related to metabolism. For 24 hours, we do glucose challenge tests, and we evaluate how they respond to metabolism. These blood sampling is done without waking up the subject. There's a hole in the wall and a long catheter, so they sleep freely during this time. And we were interested to understand some of the mechanisms related to insulin resistance, and one of the things we looked at was the free fatty acid levels across the night, across the 24-hour period. And in red, you see the sleep restriction condition, and as you can see, the early morning and middle of the night and early morning free fatty acid levels were elevated when subjects were sleep deprived. And then we also use in the protocol identical meals, so everything is controlled in terms of metabolism in the laboratory. And when we tried to understand why free fatty acids were elevated, you can think of 3 hormones that stimulates this release from the fat cell, right? Growth hormone, catecholamines, and cortisol. And growth hormone, the pulsatility, if you can remember from the first few slides, in a normal sleep condition, we have this nice big pulse. It became biphasic with sleep restriction. So the subjects, even though the amplitude was lower, were exposed to more duration of growth hormone at night after sleep restriction. What happened to catecholamines when the subjects were forced to stay awake? Catecholamines level were higher, and they weren't walking around. We keep them always in a semi-recumbent bed position, so this is not position related. This is body's physiological response. The cortisol levels, particularly the evening cortisol, which the period where cortisol is supposed to be quiescent, was increased. The morning pulse, of course, occurred earlier in the sleep restriction condition. But altogether, they contributed to increased free fatty acid levels. And also, we measured the insulin sensitivity. Like others have found, insulin sensitivity was lower with sleep restriction, and that was predicted by increase in the free fatty acid levels the night before. So basically, the higher the free fatty acid levels you had from sleep restriction at night, that made your insulin sensitivity worse in the morning. We also took their fat cell out of the body. This project was led at the time graduate student Josiane Broussard and molecular biologist at University of Chicago, Matthew Brady. And what we did is we stimulated these fat cells out of the body with insulin at different dose. As you can see, when the subjects were sleep restricted, the same dose of insulin were not providing insulin signaling. So we gave this concentration of insulin, we obtained this kind of signaling with normal sleep. So basically there were the fat cells out of the body, they were insulin resistant, and this was the first clinical study linking sleep restriction to an alteration of a molecular pathway out of the body. So how about energy balance? Let's go through quickly the components of energy balance. The major component is resting metabolic rate, right? The 70% of our energy expenditure is spent for bodily functions, our heartbeats, or we're taking breath in and out, so on and so forth. And then we also have what we call thermic effect of food. When we eat food, we spend some energy. And then this is the more variable component called activity-dependent energy expenditure. What happens when we sleep, we're not supposed to move around. We're not supposed to eat. We lose those 2 components. And our resting energy expenditure also naturally, physiologically go down. So if we were to force ourselves to stay awake, it comes with a metabolic cost. And people have done, and our group as well, studies looking at energy expenditure under very controlled conditions. We call these indirect calorimetry rooms. Everything is measured. The subject is in a confined room. And basically what was shown is that if you were to stay awake, as expected, you spend more energy. But it was in the range of 110 or 90 kilocalories in another study, so very similar. So the metabolic cost of staying awake was in this range. However, when people were offered to eat, they were eating on average, this is a meta-analysis from various studies, when they were sleep deprived, they were eating well above to their metabolic needs. So that was shifting the energy balance towards more energy intake and basically weight gain. And there are lots of epidemiologic studies that link sleep restriction to obesity. And we also measured another hormone called ghrelin that is secreted from stomach, and that basically stimulates your appetite. And when subjects were sleep deprived, at night ghrelin was increased. You had the same meal, your ghrelin levels were staying awake. You do not get the same satisfaction from the meal, and the next meal the ghrelin levels are up. So basically it stimulates your appetite when you're sleep deprived. This is also shown that when we offered them what we call ad lib buffet, buffet style food, they ate more calories, and then their ghrelin increase predicted how much they ate. So how about reduced sleep quality? Now, we talked about duration, but can we manipulate sleep quality in healthy volunteers? We've done that, and there's a lot of interest in this group also to look at slow wave sleep. So those are delta waves that most of you are familiar with. We did 2 nights of normal sleep and 2 nights of sleep fragmentation through acoustic stimulation to suppress slow wave sleep. The aim was not to wake up the subject, and this is how we did it. This is courtesy of actually CBS 60 Minutes. They came to film us back in the day for this particular study. That's Leslie Stoll's hand, but they removed the voice. So these were kind of the sounds. We were standing in the room, and we were following the EEG at the same time. And the subject, basically, we had to use various kinds of sounds called frequency modulated sounds, because after a while, brain doesn't care. It could be 100, 110 decibel. They barely move. They have microarousals, but they don't wake up. So what are the results from this study? We were successfully keeping the total sleep time, not waking them up. This is our baseline night, and these are our slow wave suppression nights, of sleep fragmentation nights. Slow wave was suppressed. Microarousals were increased. And then dream sleep was the breaks that the experimenter took because we didn't do any intervention there. So the insulin sensitivity went down, glucose tolerance went down, and sympathavagal balance was shifted towards more sympathetic activity because of all these sleep fragmentation. This was published in PNAS years ago. So basically, then there were other studies showing that if you were to manipulate sleep quality to poor sleep quality, you also have poor glucose tolerance. How about now doing the other way around, extending sleep? If we were to extend sleep over a weekend, then what we see is, that one can partially improve metabolism by this recovery sleep, 2 nights of close to 10 hours of sleep. I've asked this question all the time, but over a long term, what happens, we don't know. And when we did, we can skip this, when we did sleep extension in the real life conditions, this was our very first experiment, uncontrolled study, we did sleep hygiene instructions to short sleepers, and we were able to get extension to 1.6 hours more sleep, and then they reported less desire for high food calories, less overall appetite, less sleepy, and this is the individual changes in sleep duration. So it is doable, and currently we're running a randomized controlled trial using more objective measures, what we call doubly labeled water method, when you can do energy expenditure in the field in collaboration with Dale Schiller, who is the first person, I'm so grateful to work with him. The first human application of doubly labeled water was done by him in the country or in the world. So these are our results from that study. We're still analyzing the study, but sleep extension experiment were successful. These 6-hour sleepers, we got 1.2 more hours of sleep. So I have to rush a little bit. I see that I have only 5 minutes left for obstructive sleep apnea. Is a major health problem, and it happens when people stop breathing at night, and it costs billions of dollars for various reasons, vehicle accidents, lost productivity, workplace accidents, and it is also associated with high blood pressure, cardiovascular disease, depression, and other metabolic diseases. Basically, in men, typical presentation is with snoring or excessive sleepiness, unrefreshing sleep. Obesity is a major risk factor, and exercise. But in women, I want to make this point that they pretty much go undiagnosed. Up to 90% of those, they don't know they have it, and they've been prescribed for antidepressants, sleeping aids, because they present mostly with insomnia or fatigue-type symptoms rather than typical sleep apnea symptoms. This is an apneic event. It has two consequences: oxygen drops and arousals in the brain. And how is it linked to the metabolic diseases? One of the major players is sympathetic system. Adipocytes is a pathway. HPA axis is a pathway. And the way we treat sleep apnea is the problem is in the upper airway, and there is a collapse in the upper airway, so we basically give, through this mask, positive airway pressure to keep the airway open through the night during sleep. Another alternative therapy for some patients, not for all patients, is to use oral appliances to bring the jaw forward so we can create more space in the back of the throat. So a lot of the research has been trying to understand how sleep apnea could be independent of all of these factors, common factors, independent of obesity, independent of inflammation. Can sleep apnea directly be linked to cardiometabolic disease? One of the studies, and there is also about 2 decades of data on this, one of the design of the study that people use is to do clinical studies and adjusting for confounders, statistical adjustment, and looking at the severity of sleep apnea and how, for example, in this case, how diabetics' glucose tolerance is worsened with increasing severity of sleep apnea. And then we have done another study. We also took lean volunteers and did oral glucose tolerance test, matched controls, sleep apneics and non-sleep apneics. And what we saw is that even at an early stage, they had signs of glucose intolerance or insulin resistance in this case, even independent of obesity. And this was an last, I think, protocol I will show is a proof of concept study we've done in the lab. We basically brought, this is a randomized controlled trial, sleep apnea patients to the lab, randomized them either to a placebo tablet 30 minutes before bedtime, night after night for 2 weeks, or CPAP. Under our supervision, we applied the CPAP in the laboratory, and here you can see night after night we eliminated apnea, we eliminated oxygen drops. This is the placebo group. We eliminated sleep fragmentation to the acceptable degree, and their glucose tolerance improved, daytime, nighttime blood pressure, 24-hour blood pressure improved, and sympathetic activity improved. This was published a couple years ago. And more recent findings from this study using a simple EKG device with 3-axis accelerometer This work is done by Florent Chapoteau, who's going to speak later today, using his biosignal processing software. We were able to get into what they call resting heart rate, which is a major factor for mortality in the cardiology field. So even though these people were ambulatory during the daytime, as you can see, these are 24-hour profiles of heart rates, hourly profiles. Compared to baseline, as the treatment goes on, the placebo group is here, CPAP group is here. We got a significant marked reduction in resting heart rate, which has a lot of implications for cardiovascular risk and mortality if you were to treat sleep apnea. And this work was led by Sashmita Pamidi. Back in the day, she was a fellow. She's now a full faculty, has her own grants at McGill University in Canada. So I think with that, I hope that I was able to give you some highlights on how sleep could be linked to metabolic health. Insufficient sleep and sleep apnea can increase metabolic and cardiovascular risk. Adequate sleep duration and quality has beneficial effects on energy metabolism. And hopefully I was able to convince you that sleep is not just for the brain, but also for your body and metabolic health, and hope you're all getting good sleep. And then I would like to thank my collaborators inside and outside the University of Chicago, our trainees. We have a big team working on nutrition, sleep technicians, and metabolic assays. And last but not least, our funding agencies, particularly NIDDK, NHLBI, and our institutional support from Institute for Translational Medicine and Diabetes Training and Research Center. Thank you, and I'm happy to take questions. On time.
You did great with the time. Thank you very much. Let's take a couple of questions, but let me just ask one quick one. So this is a systemic, ultimately sleep deprivation produces a systemic effect.
Correct.
The message I'm getting is that insulin resistance is a key part of that. And my question is, how does one, through changes in sleep, basically trigger this insulin resistance?
Right. Well, one of the things we looked at, as I've shown, is the increase in free fatty acid levels. That can drive that. Sympathetic activity drives the release of the free fatty acids. And then there's also inflammatory factors that are released from the fat cell that causes insulin resistance in the periphery. This is a very good question. In the context of sleep apnea, we just got a new funding, R1 funding from NHLBI, try to really understand now at the cellular level what's happening in the mitochondria. How mitochondria substrate utilization is changed through sleep apnea, intermittent hypoxia, and sympathetic activity.
Great, very, very nice. Let's go here. Go ahead.
What are the major contributors to jet lag?
Very good. Yes. So jet lag is something that a lot of us experience. And it basically, you probably recognize that it basically takes you a couple of days to get adjusted. And we call it almost like 1 day per time zone to get used to the new environment. It's because what we call the circadian misalignment. The environment is different. Our internal clock is at a different phase. So for them to get aligned, we have to get the environmental cues at the new place that we travel to make them communicate better. So it's dissociation almost from our peripheral clocks and the circadian clock. So.
Time for another question. Todd?
It's fascinating that you said 90% of women go undiagnosed for sleep apnea. Why physiologically is that the case, or is it more of just a healthcare practice where doctors are not listening? So if men present with the exact same symptoms as women, they take one path as compared to women, or is the physiology different? Or I was just curious about what the reason is.
The clinical presentation is different. There are social factors and there are some pathophysiological factors that we need to learn more about. One of them is that women tend to present more with what we call microarousals. They desaturate less. They present with insomnia symptoms more so than being in the waiting room and snoring and being a sleepy person. They could be lean. They could be presenting with what we call REM dependence, rapid eye movement sleep dependent apneas. And the other aspects is that, unfortunately, a lot of times men are diagnosed by their bed partner. And they're pushed to the sleep labs, but the opposite usually doesn't happen often. So there are various factors there, but there are clearly gender-related differences in not only the presentation of sleep apnea, but also how the outcomes are affected. Who is more susceptible to cardiometabolic risk, for example.
Thanks. One last question in the back.
Can you comment on the gasping and also the hypercapnia, hypoxia with sleep?
Can I comment on what?
Gasping, if the oxygen content goes to
Gasping.
2%.
Okay,
yes.
right. Yes, well, gasping is a consequence of trying to catch up your breath, the recovery breath after breathing has decreased through either apnea, complete cessation, or decreased breathing. And at that point, yes, there is what we call reoxygenation from hypoxia, and there are changes also in CO2 concentration. So people are also trying to understand to what extent intermittent hypoxia, deoxygenation, reoxygenation versus changes in CO2 contributes to some of these outcomes. So both of them are contributory to physiology there and to the sympathetic activation ultimately at the end of the breathing event. Yes.
So I think we should move on. If you want to go ahead and get ready.
Yeah. Thank you.
Just one last, advertisement for Brain.
Issue.
It's very interesting that brain also seems to be impacted quite significantly. It will be interesting to know to what extent insulin resistance in brain is a part and parcel of sleep deprivation.
Very good point. There's always a circle back, right? So there are insulin receptors in the brain, and there is communication from the periphery to the brain to regulate metabolism.
Everything comes back to the brain. Sorry, I'm a neurologist, but what I say.
It does.
Great. Well, let's move on. Thank you very much for that presentation. Thank you. Appreciate that.